Us to our plenary lecture, and I'll turn the floor over to Roseanne Ford, Professor of Chemical Engineering at the University of Virginia.
Hi, good morning, everyone. It's my pleasure to introduce our plenary speaker today. Eugene Vorobiev is a professor emeritus at the Compiègne University of Technology, France. He was a full professor of chemical engineering and head of the Laboratory for Agro-Industrial Technologies from 1997 to 2021. He received his engineering diploma and PhD degree from the Kiev Technological University in Ukraine and his Doctor of Habilitation diploma from the Université de Technologie de Compiègne. His main research interests are focused on solid liquid extraction, filtration, expression, and electrically assisted separation processes. His team provided pioneering research on pulsed electric energy enhanced processing and technologies. He's published more than 400 scientific peer-reviewed papers. He's the author of 19 patents, 6 books, and more than 40 book chapters. He's a member of the editorial boards for several international journals, including Separation and Purification Technology, Innovative Food Science and Emerging Technologies, Food Engineering Reviews, and Agricultural and Food Chemistry. He was awarded the Gold Medal of the Filtration Society, and he was twice a laureate of prize of the French Agency for Ecological Transition, ADEME. Please join me this morning in welcoming our plenary speaker, Dr. Vorobiev.
Thank you very much for a kind introduction, and I would like to express my gratitude to the organizers of the InterPore conference to invite me here. It's a big pleasure for me to be here. My presentation concerns enhancement of mass transfer of biological media using electroporation of cell membranes, and I will speak about the mechanism and technological applications of this process. Here you can see just several examples of biological porous media like yeast, activated sludge, green biomass, edge, etc. Here you can see another examples like sugar beet strips, carrot slices, apple slices, spinach mash, etc. So in most cases, we have the cellular structure of biosolids particles with, of course, the elements like vacuole, cell wall, cell membrane, and there are different compounds of the cell. If we see more detailedly the structure of the cell membrane, this is the lipid bilayer surrounded by a water solution, and this cell membrane is very thin. The thickness is just 10 nanometers and even smaller, but this membrane is very dense and low permeable. Here you can see another examples, biological membrane cell tissue for plant tissue for different microorganisms. So you can see that in all cases we have a very dense cell membrane and also the cell walls, which also present the porous media. Here you can see the schematic presentation of the transport in the cell tissue, so which is intact. So the plastic transport, symplast transport, different routes. And the cell membrane can be considered like a semi-permeable membrane, which is can be traversed by some molecules, small molecules, and small uncharged polar molecules like water, but it is impermeable for larger molecules like glucose, sucrose, for ions, and there exists the special routes for cell membrane for humidity attraverse the cell membrane. Also the cell can be considered like some factory, like some plant which produces different cellular compounds which can be valorized, like vitamins, like enzymes, lipids, polysaccharides, proteins, etc. But to extract these compounds, it is necessary to destroy the cell membrane and generally destroy the cell tissue. Most often, it is done by mechanical methods, for example, by grinding. Another methods are often used like thermal treatment, heating, chemical, biological treatment. But in most cases, such treatments are non-selective. This is why it is also used innovative physical methods, which are more selected to destroy, to damage the cell and the cell membrane. And one of these methods is a pulsed electric field. So I will speak more about this treatment. Here you can see the different applications of the pulsed electric field. If we expose the cell to the electric field, you can obtain the phenomena which is called electroporation. This electroporation can be irreversible or it can be reversible. For the reversible electroporation, the cell disappear and the cell reseal after the treatment. And for the irreversible electroporation, the cell in membrane exists even after the treatment. So there are many different applications for irreversible electroporation. For example, we can cause a cell death, which is interesting for inactivation of microorganisms. We can obtain also extensive extraction. With a reversible electroporation, we can introduce the small molecules inside of the cell. We can also selectively extract the molecules and also introduce large molecules. Also, it is possible to have the cell fusion, the protein insertion in the plasma membrane. So different possibilities can exist with such a treatment. And now you can see also better the parameters of electroporation. So with lower intensity or electric field strength, we have a reversible electroporation. If we increase the electric field strength, we obtain non-thermal irreversible electroporation. And then if we again increase the electric field strength, we have also the thermal effects. Also, we can see from this graph that the diapason of the time of the pulsed electric field treatment is short. So you can see that it changes from nanoseconds to milliseconds generally. And also here you can see the cell membrane before electroporation, during electroporation, and after electroporation. So in this case, it was the reversible electroporation. A few words about the mechanism of this process. So it depends from the transmembrane potential, There exists a well-known Schwann formula which shows the relation of this transmembrane potential from the parameters of electric field, from intensity of electric field. It depends also from the size of the cell, depends from some physical factors, depends also from the direction of electric field, the time of the membrane charging, and if intensity of electric field bigger, if the size of the cell is bigger, it is more easily to attain some critical value of transmembrane potential, which is about 1 volt for the plant tissues. Of course, this formula is simplified, and other factors should be needed to be in consideration, like the orientation of the cells, like the form of the cells, like the connectivity of the cells, for example. Well, there are many applications. For example, for medical application, I will say just a few words. So electrochemotherapy, you could put the electrodes near the tumor, and then chemotherapeutic surrounds the cells, and we apply the short pulse electric field. The membrane pores appears and chemotherapeutic goes inside of the cells, and then we have the pore residue. And this is very effective treatment to treat the different type of tumor. Here you can see the melanoma was treated. At the left, you can see before therapy. In the center, it's after 1 month, and then after 6 months, as you can see that the melanoma disappeared. So this is very interesting application in medical works. And but in my presentation, I will speak about another application of electroporation. This will be for the mass transfer to extract the different valuable compounds from inside to outside of the cells. In this case, we can create the pores in cell membrane and enhance different flows, solid flow, humidity flow, filtration flow, and also increase the effective diffusivity coefficient, the permeability coefficient. How to see by eye very simply that electroporation was produced. Just to see here is the sugar beet tissue before the treatment, and here just after the treatment you can see the humidification of the tissue after the treatment because the osmotic pressure is higher inside of the cells than outside of the cells, and there is a creation of some humidity flow after electroporation. So humidification can be very easily detected. Here you can see the cell structure images after electroporation. So the onion epidermis was surrounded by some colorant, and after electroporation of cell membrane, colorant diffuses inside of the cells. You can see that the cell structure seems to be intact, and but colorant diffuses inside, so the cell membrane was damaged. And also we can see that with increase of the intensity of electric field and with increase of the time, the diffusion of colorants increases, so it takes some time for the diffusion. Here you can see another examples of microscopic studies for the red beet tissue before and after treatment and extraction. So the colorant was extracted for the onion, potato tissue. In all cases, you can see that the structure of the tissue was, it seems that it was nearly intact. Here you can see another examples for the wine grapes before and after electroporation. Here you can see the images of cross sections of grape berries before electroporation, and after electroporation you can see that polyphenolic compounds appears from inside of the tissue, and tissue seems to be intact. But of course the process is rather complex. To see this, you can see the skin cross sections of the grape berries. So you can see here the cell wall for the untreated berries. I don't know if it's possible to see the cell wall for the untreated berries, day 0, day 14, and with the pulsed electric field, it seems also that the cell wall was intact, day 0, but after 14 days, you can see that there are some changes with the cell wall, shows that it was some biological modifications of the tissue. But what is important that just after the treatment, the cell wall seems to be intact. How to determine easily the degree of electroporation? It can be done by measurement of the electrical conductivity of tissue. So we use for this cell permeabilization index or cell disintegration index, which shows the ratio of actual electrical conductivity of tissue minus initial electrical conductivity divided on the electrical conductivity of completely damaged tissue minus initial electrical conductivity. It means that for the intact tissue, Z equals 0, and for the completely damaged tissue, Z equals 1. So the degree of damage can be changes from 0 to 1. And here you can see the examples of determination of such factor Z and characteristic damage time for different food materials. You can see for tomato, apple, kiwi, etc. So in this case, we can say that the characteristic damage time, this is the time which is needed to damage 50% of the tissue. It means that when Z equals 0.5. So this is a semi-denaturation of the tissue. So like this, we can determine the characteristic damage time of tissue, and it decreases with a higher intensity of electric field. But if, for example, we fix the intensity of electric field at 400 watts per centimeter, like example, then we can see the characteristic damage time can be different for different food tissues. It is smallest for the potato tissue, just several milliseconds. It is bigger, for example, for orange banana tissue, it's about 0.1, but in all cases, the time is very short. This is why the energy consumption is very low with this treatment. This treatment is considered like non-thermal treatment. So energy consumption is many times lower than for the heating. And let us compare the time with electrical characteristic damage time. Here, the example was done for the shikari tissue, but we can obtain the same type of the graph for many other types of the biological tissue. Let us see what we have with the heating. So, for example, for thermal treatment heating, we need about 1,000 seconds to heat the tissue at 60 degrees to damage the material. If we decrease the temperature, this damage time increases. But if you just apply the rather small electric field with intensity of 100 watts per centimeter, you can see that we can decrease importantly the characteristic damage time, and also the line in the semi-logarithmic coordinates becomes more close to the horizontal line. If we again increase the intensity of electric field to 400 or 600 watts per centimeter, this line becomes nearly horizontal. It means that the temperature is not important for such treatment. We can work at the ambient temperature to damage the tissue. Also, it is not important to increase the intensity of electric field from 4 to 600 watts per centimeter, because even at 400 watts per centimeter, the tissue was effectively damaged. Let us see how to use it in technology. So we can have different feedstocks, food plants, green biomass, etc. Generally, the compounds which we would like to extract, like polysaccharides, proteins, lipids, colorants, are inside of the cell. So first of all, we need to destroy the cell to extract these compounds. Generally, it is done by mechanical, thermal, chemical methods, which are non-selective. If we use the pulsed electric field, it is more selective for the destroying of cell structure, and we can obtain then the primary extraction, which is more selective, using, for example, drying, pressing, solute extraction, osmotic dehydration, filtration, centrifugation through the electroporated cell membranes and through the cell wall cell tissue. Then we obtain the extracts and partly exhausted biomass, like, for example, cakes of rapeseed, sugar beet pulp, grape mesh, etc. Extracts can be purified and to obtain the final products, for example, the sugar, the powders, and we can also perform the secondary extraction of byproducts to obtain, for example, the protein solutions, and exhausted second time biomass can also be used in biorefinery to be transformed to obtain the bioethanol. So in this case, you can see the whole biorefinery, but it depends from the pretreatment. If pretreatment is more selective, then all downstream processes can be modified. Let us see now how the diffusion properties of tissue can be modified by pulsed electric field. So in this experiment, the particles, like examples, sugar beet, but it can be other biological particles, were treated by pulsed electric field, then heated in hot water at different temperature. From the solution of a classical diffusion equation, we can obtain the concentration ratio, and then from this equation we can determine the effective diffusivity coefficient. And here on the graph you can see the effective diffusivity coefficient in function of the temperature. And you can see that, for example, at 75 degrees, we have the same effective diffusivity for the untreated and PEF-treated tissue. It means that there is no interest to treat electrically if you heat at 75 degrees. The tissue was damaged thermally. But real interest is to decrease the temperature, and with PEF treatment. And in this case, you can see there is big difference for the effective diffusivity coefficient. The same will be for many other types of the tissue. Here, for example, for the shikari tissue, you can see also at the temperature of about 80 degrees, the same effective diffusivity for the PEF-treated and untreated tissue, and difference increases with decrease of the temperature. So this is for the diffusion properties. We can say the same about the compression and permeation properties of the tissue treated by pulsed electric field. So here the tissue was treated by pulsed electric field, and that compressed in this press. From the solution of classical consolidation equation, we can determine, which is analog to the diffusion equation, we can determine the consolidation ratio and consolidation coefficient, which is physically filtration diffusivity dependent for the tissue permeability and compressibility. So in this case, consolidation coefficient depends from the pressure, but it is also bigger for the tissue treated by pulsed electric field, and also for the bed of particles compared to the single particle. Here you can see again one other example for the mass and heat transfer during the drying. So here is the red beet tissue, like example. You can see that the sample was treated by pulsed electric field and then heated in this chamber by hot air, and you can measure the mass of the sample. It decreases, then we can obtain the moisture ratio and the effective diffusivity during the drying. The same you can see, for example, at 90 degrees. At 90 degrees, the effective diffusivity for the untreated tissue will be the same like effective diffusivity for the PEF-treated tissue, but at 70 degrees. It means that with the treatment, we can decrease the drying temperature on 20 degrees. And of course, the energy consumption will be lower, and also the quality of the product will be better if we dry at lower temperature. Well, just a few words about the modeling here in this conference. Often speak often of the model. Here it was as a sample, a dual porosity model for mass transfer in the electroporated tissue. At the left, you can see for the diffusion. On the right, you can see for the compression and consolidation. So there is a different concentration inside and outside of the cells for the diffusion, and different pressure inside and outside of the cells. So it is possible to apply the model which is resembling to the models in the flow in fractured rocks, like classical models for rocks. And then with some boundary and initial conditions, it is possible to obtain, in simplest case, analytical solution. If no, it's numerical solution. And here you can see that it was possible to predict the diffusion results by such time of the model. Here, B is normalized Briggs values, and Briggs shows the concentration of solids in the solution. So FP is the surface fraction of force induced electrically. Most my presentation concerns the technological applications. So I would like also to speak a few words about the selectivity of solid separation with pulse electric field. So in this experiment, the biological particles can be put inside of the press. We can compress the bed of particles by elastic diaphragm or by piston, for example, and it is possible to apply the pulse electric field before the treatment or during the treatment with electrodes which are permeable. So what we can obtain with it? You can see here examples for the carrot tissue, apple tissue, but they can be done for many other tissues. The treatment was applied after 1,000 of seconds. And you can see immediately there is increase of the juice yield in both cases, but also at the same moment there is a decrease of absorbance. So the juices became more clear. Also, there is a change of the bricks, the solids which were extracted. So I mean that with such treatment, we can from one side enhance the flowing, from the other side, we can influence on the selectivity of the flowing because some molecules can be retained inside of the tissue. The same you can see this example of, sorry. This example is for the white grapes. So you can see that with application of pulse electric field, there is increase of the juice yield. Here applications were done at the beginning of the process, but also there is decreasing of turbidity and absorbance of the juice. So in all cases, we have the results which are practically similar. Let us see how to scale up such treatment. Here you can see the small pilots equipment at the left for the pressing. You can see the screw press with electrodes. And at the right, you can see the countercurrent extractor, small extractor for the extraction of compounds from sugar beets in this case. So with pressing, we can immediately increase the juice yield, obtain the same juice yield at 20 degrees, like after the heat treatment at 70 degrees. And with diffusion, we can decrease the quantity sucrose in the particles, and here you can see exhausted pulp, so the quantity sucrose nearly zero, but on this other case, it is increasing of the concentration of the juice. Here is the diffusion juice. So this diffusion can also be done at the temperature which is close to ambient temperature at 30 degrees in this case. So principally, we can go from the hot technologies to cold technologies. And let us see how to do it on industrial scale. So here you can see the biorefinery based on the sugar beet. So you can see the sugar beet with tails, then the sugar beet can be sliced to obtain the particle, and existing technology to obtain the sugar. This is a thermal denaturation of tissue at about 95 degrees. And then the thermal extraction at about 70-75 degrees during 1 hour. This is very effective technology to obtain the sucrose. It exists more than 150 years. But unfortunately, non-sugars, undesirable compounds like pectins, proteins, colorants also goes to the juice because of such unselective extraction after the thermal denaturation. And this is why very complicated purification station exists in sugar factory with many equipment just to separate this impurity before juice concentration and sugar crystallization. So if you use instead of thermal treatment the pulsed electric field treatment, in this case we can more selectively damage the tissue. We can use non-thermal extraction or pressing, and in this case the quantity of non-sugars will be considerably lower in juice because this non-sugar will remain inside of the structure, and of course the purification station will be considerably lighter. Then the pulp can also be treated by high voltage electrical discharges to obtain the pectin. The tails can also be treated by pulsed electric field to obtain the juice for bioethanol production. So this is a whole biorefinery based of such treatment. Here you can see the industrial installation, the photo of industrial installation with pulsed electric field generator and with presses for such process. I will show the short video. So you can see here the treatment was 10 tons per hour. Here are the cassettes obtained after the slicing from the beads. And the cassettes goes continuously to the treatment chamber. Here's the generator of pulsed electric field treatment. And you can see the form of pulses, which is a square form. And here, this is outside of the treatment chamber. And the treatment is done in this chamber, which is collinear during the continuous flowing of the matter. Here is the treated material. And then this material goes to the screw presses for better pressing. This is the screw press. And this is the treatment. All of this is done at the end. The sugar obtained technology, we have the juice purified, then white sugar, and with new technology, you can pressing at ambient temperature, ultrafiltration, and see also the white sugar of good industrial scale experiments for the so the apples should be washed, pulsed electric field chamber, and the mesh goes to the belt press for the pressing. Here's the juice yield. You can also, to obtain small video, you can see the here there is the mesh is sample pumping. Here you can see the generator. To the belt press, here is treated, and there is this is industrial belt press pressing, and that you can see here. Just two more to use the pulsed electric field for other for freezing taving. Example of the tissue freezing of the pulsed electric is more rapid. You can see that with increase goes here, so the temperature decrease. The tissue is has texture is better preserved, and the production. So you can see here. You can see that the slices are more elastic for the carrot tissue, but the same will be for the sterilized on more than 100 factories of Mackay French fries. So nature is an excellent can be functionalized for the selective mass Biological membranes can be and this grease or selective echo extraction technologies. Thank you.
Thank you very much. The food that we eat every day. For questions, raise your hand and we'll get a mic to you via the online portal. Start off with a question. I was curious about damage time for the different fruit. Thickness of the skin on the fruit, something related to the membrane itself. Environmentally.
We determine this damage time.
Wondering, like, were there things maybe at the molecular level? Different times.
Of course, we the fruit. Fruit, sometimes it's necessary to measure. Rather easy to do. The juice, the liquid and outside. So when we treat the electrical conductivity is lower, and during electrical conductivity increases, but to damage completely, it will be the maximum value of the electric. The maximum damage tissue and initial of the permeabilization of tissue. It can be for thermal treatment, for example, not just for
this lecture. As a chemical engineer, I can relate. Different. Mother Nature now processes everything in place, almost everything. And our agriculture, you had a sugarcane plantation, you would into the soil very carefully. In Thailand, for example. Now, we'll to a biorefinery where you do a fab on the end, at the end step, the truly fantastic inefficiency system as a whole. So, of biomass so effectively, do you suggest to return some of this or some other ecosystem so as to reduce? Because right now we are 5-to-1, 10 parts of energy in engineer. I can tell you this, very.
Yes, it's not. In any case, the idea of act selectively. It means waste, but the compounds can treat then the product for other. I think if you want to, for example, there are the sugar beet, which is rich in sugar, and but they will have this pulp for should be very selectively chosen to try to do the process.
You showed one example of a trial treatment, I guess,